we're going to take a look at the evolution of populations. The first key point that we want to understand as we start to take a look at this chapter is that evolution occurs in populations. It does not occur within a single individual. So what we're actually looking at in this chapter is going to be what we call microevolution. So the term microevolution means basically a change in allele frequencies within a population. And this is something that's going to occur over a period of time. So as we begin our talk of what we're going to call population genetics, we will need to go back and think a little bit about the genetics that hopefully you learned in Bio 1. So one thing that the evolution of populations is very much based on is variation. And here we see some zebras drinking some water, and a lot of us just have this idea that zebras look the same, and they are very, very similar organisms. But if you look very closely, you will start to see some differences among the zebras, even though at first glance they do look very similar. So just to point out just a little bit to get you started, if you look right underneath the eyes, notice that that area is a little bit different on all of these. And I think you would find some more differences. I know you would find some more differences if you looked at these zebras even more. But this is just to get us thinking that yes, there is variation in a population. It's very obvious variation when we talk about the human population. But even in other organisms that seem very similar to us, there is a lot of difference there. Here's a population of ladybugs. Notice that the spots are different on these ladybugs. They're also slightly different shades of orange and red. So as we begin this study of population genetics, let's go back and talk about Gregor Mendel. He is considered oftentimes to be the father of genetics. And if you look here at the years of his birth and then death, he was a contemporary of Charles Darwin. So his work, Gregor Mendel's, was actually published just a few years after Charles Darwin published The Origin of the Species. So with both of those books and research kind of put together there about the same time, people did start to put the pieces together about how evolution might actually be occurring. Remember that Gregor Mendel, he did all of his research at a time when they had no idea what chromosomes and DNA were. So he was really working strictly on numbers, and he worked with pea plants, and he looked at a lot of different traits with pea plants. And he did propose two fundamental laws of inheritance, which are the law of segregation, and then also, and hopefully these are coming back to you, the law of independent assortment. Both of these laws really have to do with the fact that the gametes, or the egg and sperm, which are involved in reproduction, those are produced through a process called meiosis. And meiosis, which is covered in chapter 13, really is the physical, biological basis of that law of segregation and the law of independent assortment. Even though at the time, Gregor Mendel didn't have any idea what meiosis was. In fact, at this time, they couldn't see cells in a microscope either. So if we go back and just talk about the basis of his laws and make sure we're all on the right track for talking about genetics, we want to refresh our minds with what we mean when we use the term allele. Allele is basically a version of a gene. And when we say that it is a version of a gene, basically it has a slightly different DNA sequence. And so this slightly different DNA sequence, what that ultimately means to the cell is that we're going to have slightly different products produced from that DNA. So the protein sequence is going to be a little bit different, and then that protein is going to behave and participate in reactions perhaps a little bit differently than other variants of that same gene. Now, these alleles are located in the same location, or what we call locus, on homologous chromosomes. So that's another term that we want to familiarize ourselves or refamiliarize ourselves with, hopefully. Homologous chromosomes, this is basically a chromosome from mom and a chromosome from dad. And these two chromosomes basically contain the same kind of information for our cells, but they may very well have different alleles on them or different versions of this information. So Gregor Mendel worked with pea plants, and one of the things that he looked at quite a lot was flower color. So here we have mom is going to transmit or pass on a purple color to the offspring, and dad is passing on the white color to the offspring. Keep in mind that as humans and most animals, and what we're going to be focusing on, we are diploid organisms. And since we are diploid organisms, that means that we have 2 copies of all of our chromosomes. So those 2 copies, those would be the homologous pairs or the homologous chromosomes. Again, one from mom and one from dad. And we would expect to sometimes get the same version of those 2 genes. Sometimes we would expect to get different versions. So we also need to make sure we understand the difference between a genotype and a phenotype. And again, hopefully this is all review for you, because I know I'm going through it very quickly. If it's not a review, then you really need to go back and take a look at chapter 14 very extensively to get the foundation for all of this. But the genotype is basically the genetic makeup of an organism. And so the genetic makeup is really just going to tell us what alleles happen to be there in this organism. And so if we're naming the alleles different alphabetical letters, this might be something like big A, or big A, little a. Something along those lines would be the genotype. The phenotype, on the other hand, this is what an organism looks like. It may be something that's not as obvious or easy to see, but basically this is the result of the genotype and possibly some interactions with the environment. Okay, so we've got alleles, and they're going to be represented by single letters. We've got genotypes, which would tell us the genetic makeup, or which alleles are actually there. And then we also have phenotypes, which are the appearances and the looks of the organisms. Let's go to our genotypes. If we're just talking about an A gene, for example, we've got 3 different genotypes possible here. Notice that the top one and the bottom one that I've written, those have 2 copies of the same allele in them, either 2 large A's or 2 lowercase a's. So both of those would be what we would call homozygous. And the key to this is in the first part, homo, because homo means same. So what we're saying is that we basically have 2 of the exact same allele present in those 2. And we could call this top one homozygous dominant, because the capital letter is going to represent the dominant allele, and we could call the bottom one homozygous recessive, because the lowercase represents the recessive allele. The middle one here is what we would call heterozygous. And we call this heterozygous, again, the key is in the beginning part of the name, because hetero means different. And notice that you have 2 different alleles or letters there. You have a capital, and then you also have a lowercase. Okay, if you have 2 heterozygous parents and you cross them with each other, the first thing that has to be considered is we have to consider what kind of gametes these parents could possibly pass on to their offspring. Well, gametes are what we refer to as egg and sperm, and gametes are always going to be haploid. So a gamete is going to get 1 allele of each gene. So there would only be one A in each egg or sperm produced by these. So if this was going to be our female right here, she's going to be making eggs. Half of them are going to get a capital A, and half of them are going to get a lowercase a. That's because she has both. Okay, so half of the time it will be one, half the time it will be the other one. And if we consider that this one over here is going to be the father or a male, same thing for him. Half the time he's going to pass on the capital A, and half the time he's going to pass on the lowercase a. Now, since it's going to be random fertilization, we don't know which egg is going to get selected, and we don't know which sperm is going to get selected for that fertilization. So we have to consider all possibilities. This is where the Punnett square comes into play. So with the Punnett square, we would write the eggs on one side. So I'm going to write them here. And we would put the sperm on the other side. So I'm going to put those across the top. And then to fill in the boxes, we look directly across and directly above. And I said boxes, so we can go ahead and divide this into 4 boxes here. So if we do that, the first box gets 2 capital A's. This box gets a capital and a lowercase. Again, I'm looking to the side, and then I'm looking directly above and looking at what I have on the outside of the Punnett square. And then for this bottom box, and then over here. So as we look at these, the genotypes are the letters that we see written inside the squares, and then the phenotype would be what they actually look like. If we consider that A is dominant, to have the A look, all they need to have is at least 1 capital A. So this would include the first box, this box, and then also this one. So we actually have 75% or 3/4 of our offspring that have the capital A look, or the dominant phenotype. And then for the little a look, for that one, the only way that it's going to look like that is if you have 2 lowercase a's. There's only one box that has that, so we have 25% or 1/4 of them actually looking recessive. All right, so as we move on and start talking about genetic variation, genetic variation is really what population genetics is all about, or what the evolution of populations is all about. So by genetic variation, this is basically variation in the genetic material. So we have to keep in mind that that would be DNA. So we're talking about DNA differences. And this specifically is DNA differences among a population, or in other words, between individuals within that population. Keep in mind that the DNA is made up of 4 nucleotides. We have A, T, C, and G. And those nucleotides, we have billions of them if we're just talking about human individuals. And so there could certainly be differences between my nucleotide sequence and then yours. There will be differences between those. And we can also have differences within the actual genes. These nucleotides here, there's 4 of them. We said that we have billions in humans. The numbers are a little bit different if you're talking about a different organism, but there's still going to be lots of nucleotides there. And then different sections or groupings of those nucleotides are going to be what we call genes. So a gene will be, we'll say, a group of nucleotides that basically specifies, I'm going to say specifies how to make a product. Okay, and by that product, most of the time we're talking about proteins. It might be an RNA molecule. So there's a little bit of differences there. But we need to understand when we're talking about genetic variation, it is the DNA that we're talking about. It is the individual nucleotides that could be different. And those different nucleotides may lead to different genes, which would then have slightly different protein products. Okay, so variation within a population, that's what we're talking about. We also want to keep in mind that there are different types of genes or different types of characters, appearances, phenotypes that you can have. When we talk about discrete characters are basically ones where you have basically one gene affecting one thing. And so this, if we think about Mendel's experiments, he had one gene and it affected flower color. Okay, so what you inherited based on that one gene determined whether the flowers were purple or whether they were white. If we talk about some human characters, widow's peak is a good example. It's one gene involved in determining whether you have a widow's peak or you do not have a widow's peak. So when we're talking about discrete characters, it's very easy to work out genetic crosses. And there's not going to be a whole lot of variation a lot of times in the population because there just aren't quite as many possibilities there. Now, if we look at the second type of variation that we can have within a population, this is quantitative characters. And this is really a lot more of the variation that we see. Quantitative characters, these are basically things that vary along a continuum. And when we say that they vary along a continuum, this means that there's not, say, a short and a tall. There is any possible height that you can imagine. There's not a specific, two specific weights that we can be. There's a whole range of weights. If we talk skin color, there's not really a white and there's not a black. There's any shade of tans and browns involved there. So quantitative characters, there's a lot more possibilities. Basically anything that you can imagine is going to be varying. If you can just see like a range of individuals, that's going to be a quantitative character. The reason that they vary along this continuum is that these are controlled by many genes. Okay, so the genetics for these are certainly not as easy to work out for genetic crosses. They're near impossible a lot of times to work out just because of the number of genes involved. Not only do you have a lot of genes involved, but you may also have a lot of different possible alleles involved for each one of the genes that we're talking about. So we want to be able to talk about genetic variation and how much genetic variation we have within populations. Because keep in mind that how much genetic variation we have, basically this is going to control the ability of a population to evolve. So it's really very critical that we have a lot of genetic variation within a population. So if we talk about how we measure this, we can measure this at the whole gene level. When we measure it at the whole gene level, we basically are looking for how many loci. And remember that a loci, or loci is plural for locus, and this would be specific locations on the chromosome. So we would say how many loci or genes are heterozygous. So this would be within a population. We want to know how many, on average, do you have that are heterozygous. So we call this the average heterozygosity. Okay, so if you had 100 genes, and if you went around and surveyed a bunch of people and you found on average that they were homozygous for, say, 78, and so we'll say, what we say, homozygous for those, and that would leave us 22 that they're going to be heterozygous for, then we would say that the average heterozygosity is going to be 22% in that case. Okay, so that's one way to measure genetic variation within a population. Just go around, do a survey, look at how many genes are present in the homozygous condition and how many of them are in the heterozygous condition. A second way to do this is to look at it at the molecular level. So this is looking at it a lot more specifically. This is actually looking at the DNA. So here we are comparing actual nucleotide sequences with each other. Okay, so here we would go along and we would actually compare the A, T, C's, and G's. To do this, you would need to do a lot of sequencing of the organism's DNA to get a good idea of how they compare. To get some idea of numbers, gene diversity, or whole level, we'll say average heterozygosity is another way to say it, is about 14% in humans, but the nucleotide diversity is only 0.1%. Well, it may seem strange to you that the gene diversity is 14% and the nucleotide diversity is so low at 0.1%. But keep in mind that we don't have as many genes present. And so when you have some that are present in a heterozygous form, that's going to have more of an impact on that percentage number that you're getting. So that's pretty good, the percentages that we're seeing here. If we just say the 0.1%, nucleotide diversity or molecular level diversity is a pretty good source of genetic variation. It's certainly plenty of genetic variation for a population to evolve in response to natural selection and some other things that we will discuss.